In this video, we're going to talk about basic probability and why sometimes things don't happen as we expect. So how can we predict the chance of inheriting genes and the chance of certain traits appearing? Well, we know that when there's two parents, it's not a guarantee that you get one parent's gene or another parent's gene because it's 50% of one parent's DNA and 50% of another parent's DNA. So here, for example, I have two flowers, a 4-petal flower and a 6-petal flower. And if they reproduce, what's the chance that the child will get one gene or another gene, right? Or show one trait or another trait. As you know, there's something called dominant and recessive, right? So dominant gene is the gene that is going to be used, and the recessive gene is only used if the dominant is not there. So we're going to pretend 4 petals is the dominant gene, and we're going to use capital F to represent 4 petals. And we're going to pretend that 6 petals is the recessive gene, and we're going to use lowercase f to represent recessive. So the first vocabulary words here is homozygous. Homozygous just means that the two genes are the same. Now you might be like, how come there are two genes in this one parent? Well, this flower had parents itself. And so this flower had a mother and a father, right? So there's two genes, one from mother, one from father, right? So if the plant is 4 petals and we said 4 petals is dominant, this parent, it could either be homozygous, where both of them are capital F, right? Because capital F represents 4 petals. Or the parent can be capital F, lowercase f, right? Heterozygous, right? Where they're different. But even though they're different, which one do you use? You don't use both the genes. You only use the gene that's dominant. That's why capital F, and capital F, lowercase f, both of these will have the same trait because the recessive gene is just not used. And in this one, there's no recessive gene. So these things are called alleles. Alleles means the same gene, but a different variation. So for example, both 4 petals and 6 petals, it's a gene telling you the amount of petals, right? But they are different alleles, right? So the capital F represents 4 petals, right? While the lowercase f represents 6 petals. So they're the same part, right? But it's variation of that part. So the part is the petals, right? But the variety is how many petals. That's why they're considered alleles. Okay? All right, so let's go ahead and look over here. If 4 petals is common in nature, that's called the wild type. So wild type doesn't have any special meaning. Wild type is just talking about what is seen in nature a lot. Okay? All right, so now that we understand this basic about how the dominant trait can show up as long as one dominant gene is there, while the recessive trait can only show up if both of the genes, both of the alleles are recessive. Now let's go ahead and go into the prediction part, right? Let's pretend we know that the parents' genes are this. So capital F, lowercase f, and lowercase f, right? So looking at this flower, I would have no idea whether it's this one, heterozygous, or homozygous, right? I would know for sure that this one has to be homozygous because the only way to show the recessive trait is by having 2 recessive alleles. But for this one, I would have no idea. So how do we get this? We'll talk about a test cross in the next video, but let's pretend we somehow know this flower's exact genes, right? So how do we make the prediction? Well, you use a Punnett square. So Punnett square, you just put the genes of one parent on one side and you put the genes of the other parent on the other side. Okay? And so you can see I put the capital F here. So this parent, it can contribute this capital F. But what else can this parent contribute? It might instead contribute this other F, which is a lowercase f, right? And in the other parent, it's the same thing. Maybe it gives this lowercase f, or maybe it gives this other lowercase f. In this situation, because this parent is homozygous, it doesn't matter, right? Because both of them is going to be a lowercase f, right? But in this parent, it matters because it's heterozygous. So if we put it out, now let's look at the 4 boxes. 2 out of the 4 boxes have the capital F, right? So only those with the capital F will be able to show the dominant trait. So 50%, 2 out of the 4, 50% is going to be 4 petals, which is the dominant trait. Only 2 out of the 4 have all lowercase f, right? Remember, if there is a dominant trait, dominant allele there, the recessive doesn't get used. So 2 out of the 4, 6 petals, right? Because 2 out of the 4, 50%, have only recessive alleles. Okay, so that's it. That's how you do the basic Punnett square. So you try, right? So if you have one parent that's capital F, and you have another parent that's capital F, lowercase f, what's the chance of 4 petals? What's the chance of 6 petals? So I don't even actually need to do the Punnett square for this, because I could see that this parent, no matter what, it's going to contribute a dominant allele. So does it even matter what this parent contributes? It doesn't, because as long as the child gets one dominant allele, that's the allele that's going to be used. That's the gene that's going to be used, right? But you could do a Punnett square like I just did, just to see for yourself as well. Now, some of those children will be carriers. Carrier meaning that they'll have the recessive allele, but they won't actually be able to use the recessive allele because they'll have the dominant allele there. All right, now let's move on over here. What if I want to know the chance of 2 traits, right? So I'm going to multiply my probabilities then if I want to know the chance of 2 traits. So let's pretend green is dominant, red is recessive, and we're going to use the same allele from before. So we're going to use the 4 petal or 6 petals. So let's say one parent is capital F, capital G, lowercase g, and the other parent is 2 lowercase f's, 2 lowercase g. So I'm going to do the Punnett square for each one individually, right? So G, I'm going to do it over here. Notice how I only did the capital G's, because if I figure out which boxes have capital G, then those are the boxes that's going to have the dominant trait for sure. Any boxes without the dominant trait, dominant gene, sorry, will only express the recessive. So 2 out of the 4 are going to have the capital G, so it's a half chance of green, right? Am I done? No, because I want to know the chance of 2 traits, right? So let's go ahead and look over here. I'm going to do the other one, right? So in this one, all of the boxes have the dominant allele, like we just discussed on this side, right? Because one parent is 2 capital F's. So it's going to be 4 out of 4, which equals to 1 out of 1, which is 100%. Basically, all of them, all the children will definitely have 4 petals, right? That's the dominant allele, and the child has a 100% chance of inheriting at least one dominant allele. All right, so now that we know the probabilities of each individual trait, now we're going to use multiplication if I want to know both, right? So the chance of green 4 petal would be 1/2 times 1, right? Because 1/2 is green, and the chance of 4 petal is 1. So 1/2 times 1 would just be 1/2. So 50% chance of getting a green 4 petal, right? What's the chance of a green 6 petal? So we know that having green is a 50% chance, right? But having a 6 petal is 0% chance. It's impossible to have a 6 petal plant here. So 1/2 times 0 is 0, otherwise 0%, right? So because you want green 6 petal, the green part might be possible, but the 6 petal part is not possible, right? So that's why it's a 0% chance of having both of these things. Now the chance of red 4 petal would be 1/2, because red is the opposite of green here. So 1/2 times 4 petal, which is 1, so that's 50% chance as well. Now what if you want to know the chance of OR though? Let's say you want to know OR. In OR, you're just going to add. So here's an example. Green or 6 petal. In the situation where it's green or 6 petal, right? So the chance of green would be 1/2. The chance of 6 petal would be 0%, right? So 1/2 plus 0, it's a 50% chance that you would have a flower that is green or 6 petal. You just add the probabilities together. When you say or, it means you don't care, right? You don't need to have both conditions. Here, it was 0% because you needed both conditions, you multiplied. But when you say or, that means you don't care which one you get. So you have a 50% chance of getting green. Great. But you also have a 0% chance of getting 6 petals. So you add half to 0, it's still half. Okay. All right, moving on. So what we just did was called complete dominance. Why is it called complete dominance? Because as long as you have one dominant allele, that's the only allele you use. But that's not always the case. So over here, we have pretend stripes is dominant, right? So complete dominance, we don't know what the other gene is. As long as the dominant is there, that's the one that's used. And that's the whole point of the Punnett Square, right? By the way, this is supposed to be a zebra. Let's pretend the spots is recessive. In incomplete dominance, right, both genes are used, but partially. So you're going to see a new trait, like a mixture. So you see these don't really look like stripes and they don't really look like spots, right? Because both genes are used a little bit. And so you kind of see a mix of the genes expression. And finally, codominance is when you see both genes being expressed. So you can see the zebra has both stripes and spots. So that's codominance. Both of them are being used. Let's look at this example 2 together. I'll give you a moment to look at it yourself. Pause the video here. Okay, so example 2. Why is this showing complete dominance? Because red is dominant, and that's the only gene that we see being expressed, right? So it's fully red. This one is incomplete dominance. Why? Because this is a new trait. It's not red and it's not white, right? It's pink. It's a new trait. So it's incomplete dominance because you have a mixture. Both genes are kind of used, but they're not fully used, right? The last one, this is showing codominance. Why? Because you see both red and white in the flower, right? So codominance. And the last term that we're going to go over is polygenetic inheritance. Polygenetic inheritance means that there's more than one gene for one trait. So in this whole Punnett Square thing, we kept saying how there's one gene gives you one trait. But a lot of times that's not true. So you can see here, 1, 2, 3. Maybe there's 3 genes that controls your height, right? Maybe one gene controls like how long your body is, another gene controls like how long your neck is, right? So all of these together would add together to be your total height, right? So polygenetic inheritance, there are some traits where it might be more than one gene giving instructions to add up to the total trait that you see.